Bacterial genital tract infection introduction. People infected with chlamydia often have no symptoms, therefore are often unaware they are infected and may not seek professional health care. Approximately 50% of men and 75% of women. Clinical syndrome, urogenital infection, Chlamydia trachomatis, biovar, trachoma. Female, asymptomatic 80%, cervicitis, arthritis, salpingitis, postpartum fever, increased rate. Premature delivery, ectopic pregnancy. In male, symptomatic 75%, arthritis, dysuria, and biuria. Cause of non-gonococcal arthritis from 37 to 50%. Common cause of post-gonococcal arthritis. This clinical syndrome of urogenital infection in male and female. Laboratory diagnosis of chlamydia. Number 1. Culture endocervical specimen or secretion, specimen from vagina, urethra, urethra can be cultured on McCoy cell, which are treated with cyclohexamide to inhibit metabolism and increase the sensitivity of isolation of chlamydia. These are incubated at 37 Celsius for 48 hours. The monolayer cells are examined by direct immunofluorescence microscope to see the intracytoplasmic inclusion. This special form for explain infection with chlamydia, intracellular cytoplasmic inclusion. This explained by direct immunofluorescence microscope. The sensitive and specific of this method is 80%. Number 2. Direct immunofluorescence antibody and enzyme-linked immunoassay. Number 3. Molecular method by using PCR technique. Number 4. Serology, serum antibody due to genital infection and less common trachoma are valued during and after acute infection. Gonorrhea, caused by Neisseria gonorrhea, second only to chlamydia infection in the number of reported cases. Gram-negative Diplococcus that can be cultivated in chocolate and blood agar. The organisms are found on or within the leukocyte in the urethra pus. Most are susceptible to cold and drying, and hence due to survival outside the host. This is general characteristic of Neisseria gonorrhea and gonorrhea. The most symptoms of gonorrhea in women: bleeding between periods, creamy or green pus-like or bloody vaginal discharge, bleeding during menstrual period, irritation of the vulva, lower abdominal pain, painful intercourse, painful urination, burning sensation, rectal infection, throat infection. The disease can spread into the fallopian tube, resulting in pelvic inflammation disease, which can cause infertility in up to 10% of infected women and ectopic pregnancy. Symptoms of gonorrhea in men: creamy or green pus-like discharge from penis, painful urination, burning, rectal infection, conception, creamy pus-like discharge, itching, painful bowel movement with blood in feces, rectal bleeding. Diagnosis: laboratory diagnosis of gonorrhea. Number 1. Staining biological sample. Direct for bacterium is carried out by placing on a slide a sample of the discharge from the penis or cervix and stain. Detection of bacterial bacterial gene or nucleic acid DNA. Detection of bacterial gene or nucleic acid DNA test is carried out using urine swab to detect the gene of bacteria. This test is often more accurate than culture. Number 3, culture. Growing the bacteria in laboratory culture involve placing sample on culture plate and incubated up to 2 days to allow bacteria to grow. Collection in women. Pass sterile cotton swab into the endocervical canal and genital rotate the cotton swab against the endocervical wall to obtain the specimen. In the cases where gonorrhea is suspected, before inserting the swab in anus media, this transport media, if possible, inoculated into culture media, lab and transport the laboratory as soon as possible. Culture include specific media, include modify New York City or Thayer Martin media. The specimen is inoculated and incubated overnight at 37 Celsius. For isolation of Neisseria gonorrhoeae, they will produce a small rice gray shine colony. Working oxidase positive and gram stain for Neisseria gonorrhoeae that appear as gram positive cocci. This is a general characteristic on culture media. The shape of colony on culture, small rise and gray shine colony, and working oxidase positive and gram stain. The other pathogen for genital tract include syphilis. Syphilis is a chronic systemic sexually transmitted disease caused by spirochete Treponema pallidum, subspecies pallidum. Most cases are caused by sexual contact with lesions that occur in primary and secondary stage. According to late stage of syphilis, consider non-infectious. Lab diagnosis for Treponema pallidum include dark field microscope for detection of motile of spirochete. The specimen should be examined as soon as possible after the specimen is collected. According to other methods for diagnosis include serum for RPR. Other infections include Chancroid caused by Haemophilus ducreyi. It is a fastidious gram-negative coccobacilli, a major cause of genital ulceration in tropical countries. Another early symptom is dark or light green shear. Chancroid start as erythematous papular lesion, which break down into painful bleeding ulcer with necrotic base and rigid edge. It increases the risk of HIV. The laboratory diagnosis of chancroid caused by Haemophilus ducreyi. Specimen should be collected from base and margin of ulceration, followed by cleaning with saline swab. They are gram-negative coccobacilli. Culture is required. Blood or chocolate agar contain X and V vector. The recommended media is chocolate or blood agar, but contain X or V vector that activation for Haemophilus ducreyi and prevent growth of other types of bacteria can found in specimen. Collection and transport of urogenital specimen, transport media for urogenital specimen, and collection of a specimen from men. The area around the urethra is clean. There is a message from above downward, then swab and collecting the discharge. A slide is made by rolling the swab on the slide to avoid damage the pus seal, because this bacteria can found in intracellular pus cell between the loop of nucleus. Slide is made by rolling the swab on the slide to avoid damage pus cell, dead white BC, to prevent damage white BC between the bacteria intracellular between loops of nucleus.